Amy is a computer science PhD student at UC Berkeley and Bayer, advised by Peter Abiel, and a part-time researcher at Prescient Design. Previously, she was a part-time student researcher at Google Brain and machine learning engineer at InCitro. She completed her master's in computer science from the University of Toronto and her undergrad in honors science from the University of Waterloo. Her research interests include multimodal generative modeling for protein engineering and broadly bringing task generalization capabilities from vision language models to biology. Nathan is a principal machine learning scientist and group leader at Prescient Design Genentech. At Prescient, his lab focuses on developing and applying machine learning methods to protein design. Nathan also leads Genentech's efforts to accelerate large molecule lead optimization. So without further ado, Amy and Nathan, take it away.
Awesome. Thank you so much. Thanks for the invite. And to anyone who celebrates, hope you're having a good ICLR Eve here up into the deadline coming up tomorrow. Cool. Yeah, so very excited to give a talk with Amy today and tell you about our work on tokenization and compression in the protein sequence and structure space. So I am going to give kind of maybe what is hopefully a little bit of a surprising intro and motivation to this work. I think it's probably surprising to nobody that we see protein design as a perfect machine learning problem. And as a trained physicist myself, you can certainly kind of find the protein structure representation. You can kind of look at that and be either very, very excited as a physicist and kind of think about all of the cool interactions and things that you can learn about and describe on the molecular level, or you can be like me and maybe not have quite so much hubris and be a little more humble about it and think there's a staggering amount of complexity there. And we actually have to be really, really careful with how we treat these kinds of other levels of granularity or other kinds of representations that we use for proteins. So we do want to learn from all data that we have available. We don't want to just have to stick with sequence databases if there's other data available, like there is in the form of crystal structures and function annotations and other kind of stuff. But we also know from just the limitations of structure solving and kind of what it means to solve a crystal structure, that if we just take that data as is, we're imposing really strong and often incorrect priors during the design process. In general, if we're treating crystal structures, we might also make some choices on the machine learning side that can really limit the expressivity of the types of models and architectures that we're dealing with and the types of data sets that we're able to ingest and work with as well. So we want to take a very expansive view of how can we learn from all of the data that we have available to solve this very, very hard combinatorial design problem of generating, and designing proteins that have interesting properties. So the next couple intro slides are adapted from my friend and colleague, Sam Stanton. We've had a lot of fun conversations about this kind of sequence versus structure debate, or more broadly, just thinking about different types of information that we can get in the protein space. And so these couple slides are just a summary of kind of where we come at the problem from. So of course we have annotations and labels from biochemistry and molecular biology that might tell us something about what a protein does, what pathways it's involved with, where you might find it in a cell, all of that kind of good stuff. We've got structural biology and structural information that are solved via maybe cryo-EM or other things that give us a static picture. So a very, a very specific kind of biased view of what a protein might look like. And then we have simulated data and taking things that we know from physics to try to maybe augment these data sets, simulate motion of a protein or other biomolecules, and get some dynamical information about how they might look in time. There are other design approaches, but broadly speaking, there's a lot of discussion in the literature and just like in the community about structure-based design versus more black box approaches and sequence-based design. We know that when you take a kind of structure-centric approach to design, this can work really, really well when you're working with pretty generic proteins that have some kind of highly symmetric structure, when really strong inductive biases are helpful for your problem setup. And for those reasons, these kinds of approaches tend to be very highly problem specific. So if you've got a setting where crystallization is a good representation and an accurate one of the types of proteins you care about, then you can be very successful there. In general, if we want to work more kind of flexibly with proteins that might themselves be more flexible or disordered, or kind of not fit this much more restrictive space that covers kind of only a sliver of the design space we're interested in, then we want to work with models that maybe have weaker inductive biases and are more general and expressive, and can handle larger spaces of design. Why? Why is that true? This is kind of just a consequence of how we treat priors, both when we're designing model architectures, just generally when we're approaching these problems. We can view kind of imposing a crystal structure or using crystal structure information as a very strong prior. So it's possible that if we just use sequence alone, we might end up with a prior that is too vague, and so we're not sample efficient. So for instance, if you really want to predict the fold of a protein, throwing out all your crystal structure data, probably not the most sample efficient way to get at that. But likewise, if your static structure imposes a really, really strong and specific prior and it is wrong, you will converge quickly, but you'll converge to the wrong model. So any cases where your static structure is not a good representation of the biophysics and what's physiologically relevant to your design problem, then you end up in a very, very challenging situation to get out of. So our motivation is kind of towards approaching this problem of how do you learn representations in a joint space that covers the different levels of granularity that we can describe biomolecules by, enabling us to train and learn from all data that we have available. So we don't want to just throw stuff out, but we also want to kind of be careful and relax some of these very strong priors that certain data modalities might impose on us. So that's kind of the big picture of why we're interested in these sort of joint latent spaces. And then once you have such a thing, that enables all kinds of cool downstream applications. A lot of this work that we'll talk about today was motivated by ideas related to cogeneration and generating in a joint latent space. So we'll talk a little bit about that. You could also do things like search, representation learning. Along the way, we kind of encountered some issues, and Amy did some amazing detective work and ended up doing an entire project along the way to enable cogeneration. So that's what she will talk about later in the talk. So again, the key idea here that we're motivated by is we don't want to throw away data if we think there's some signal that we can learn from it. But we also don't want to assume that just kind of focusing on one modality just because it was really hard and expensive to acquire, that doesn't mean that we're necessarily just guaranteed to get better performance on things we care about or unlock new useful capabilities. So this specific question, again, we're kind of motivated to see if we can learn a joint latent space or unlock a joint latent space that allows us to do things like cogeneration. And we know that it's very possible to do this for separate modalities or separate types of data. So you can train a diffusion model to generate structures. You can learn the distribution of structures. But when we're actually designing and making proteins, this is kind of a trivial fact, but something that is maybe misunderstood, we are never ordering a protein structure, right? We're always specifying it by the sequence. So the sequence is actually the piece that we need in order to, order DNA and express a protein and do an experiment. So anything else we're generating alongside has to be kind of with the aim of generating different, better sequences. So that leads us to this joint distribution and kind of what we can get from that. I think it's probably pretty straightforward to understand why a joint distribution and cogeneration is a very natural sort of framework. So probably most folks are familiar with this kind of multi-step approaches where first you might just generate a backbone structure with one model and then do inverse folding to kind of decorate that backbone with the sequence that you hope folds into that shape. You could do the reverse and generate a sequence first, and then try to predict what its fold will look like, what its structure will look like. And so that's kind of these marginal and conditionals here of trying to generate the apple tree. Or, of course, if you know you want an apple tree to start out with, why not just ask for the apples and the tree together in the most coherent way possible? So that's kind of why these joint latent spaces are interesting. Again, something that is super interesting to me is how we learn from all data that we have available. So if we just think about structure prediction models or even cogeneration models, but ones that are restricted to the structure space, then we've got this very, very small, very biased data set that we're learning from of crystallizable proteins in the PDB. This is very small compared to the sequence databases that we have available, and incredibly small compared to the internet scale data that frontier or foundation models are developed with. And then finally, it's probably not a surprise that we think large pre-trained models that learn from multiple types of data or multiple data modalities can potentially be bigger and better than the sum of their parts. So we want to be able to capture that in a single big model. With that, I will do a quick handoff to Amy to tell you about kind of the motivation for CHEAP and then what we did.
Cool. Okay. All right. Let me just share my screen real quick. Right. So hopefully this is shared. Yeah. So as Nathan mentioned, a lot of our sort of motivations for this work came from thinking about the desire to sort of model sequence and structure as one joint modality, and also leverage a lot of the intrinsic biases that's been captured in these large models. So that's sort of following in line with what we've been seeing in other parts of ML, right? And so one of the first places we started to look at for this was in protein folding models. We, in terms of thinking about what are the most foundation model-esque things that we have available in biology, these are one of the most capable ones. And so as a bit of a refresher, so in AlphaFold 2, we have this explicit retrieval step that gets processed. Structural features are learned from these sequence latents, and then we generate these structures. And one second. So what we use in this work is mostly ESMFolds, and ESMFold, for those of us who are not familiar, is very much like AlphaFold 2, but replaces this retrieval step with this language model. And so one of the sort of starting points for this project was I was looking through the code base for ESMFolds, and I noticed that there was a line that was just initializing the pairwise contacts to zeros. And so in the original preprint, there was a lot of sort of emphasis on the fact that these attention mechanisms in ESM2 captures contacts, and that basically can substitute these contact representation inputs into AlphaFolds. But it turns out that actually, empirically speaking, in the code base, you don't actually need like the attention per se. You can just initialize it as zeros. And I think this is a little bit counterintuitive because you would think that the structural information is very very complex, and considering the fact that you have so many trunks, you would think that the information is maybe not so compressible, but it means that actually because this is initialized as zeros, it means that actually all of our information is basically stored in this sequence representation, because at inference time, this is not actually being used. And so it means that actually all the information about the structure here is actually just stored in this fairly condensed representation. And what this means is that if we can characterize this distribution, right, be it generation or search or whatever, when you're training this model, it means that you are able to get all the information of the structure, assuming that you trust in ESMFold's prediction. And to get that representation, all you need is a sequence input. So this was one of the starting points of what we wanted to do. And you can sort of think of this sequence representation as this joint representation of sequence and structure, right? So in ESMFold, you have the sequence that goes through all the layers, and then eventually you get the structure out. And so in the middle here, because this representation only requires a sequence to obtain, but it contains all the information for structure, right? So if we can basically characterize this, then we can think of it as a useful distribution for search, for generation, for representation learning broadly. And yeah, so one of the first starting points was we tried to train a diffusion model on this space. And I think the results were encouraging, but there was some space left for improvement. So you can see that this is a very early version of this, and was actually presented at the ICML workshop this summer. But just from training on UniRef alone, from just sequence data alone, we are actually able to learn these folds, even though there was no PDB input whatsoever. So that's kind of encouraging. But you'll also notice that if you color this by PLDT, it's not like, not that PLDT is the most reliable metric in the world, but it's some ways away from natural proteins and some baselines. So, oops. So we want to think a little bit about what's preventing the model from learning more. And so a few hypotheses that we had here. One is that the training data, because of the way that we were, the fact that attention scales quadratically, the max length we could have was 128. Also, in image literature, we know that high resolution image generation is usually a lot harder. So here we have these images of 1024 by 1024 resolution. So that's high resolution, and you'll see that the faces are not always great, right? And so yeah, so the latent space we're dealing with here is actually quite a lot bigger. And additionally, one of the assumptions in diffusion modeling is that the input space has to be Gaussian distributed, and that might not necessarily hold. So these are sort of some of the questions that we had going into CHEAP. And so now I'll talk more about the CHEAP paper. So one of the first things that we looked at when we started to debug this was taking a look at the values of ESMFolds. And what you see here is, here you have the hidden values by the channel dimension, and you have the input dimension along the sequence, and along this vertical axis, you see the magnitude. And so there are certain channel dimensions that no matter what position you look at, and no matter what input you give it, it just has a really, really large value. So from a generation perspective, this means that, well, it's not Gaussian distributed. This is a very, very wacky distribution. But it also has, oh, sorry, yeah. I also wanted to mention, yeah, so it's not just for that one layer. This is sort of an issue for a bunch of different layers. And so it's not just like a per latent space issue, but sort of an issue throughout the entire model. And this massive activation actually starts off from the ESM2 language model that was sort of like the first half of the model. And so here you see the top 3 activation values for each layer. And in gray, you have the median activation value. And so you see is that for the 3 largest values, they are quite a lot bigger than the rest of the values. And this basically starts out, somewhat manageable, and then it kind of just grows and grows through the ESM2 layers. And this is not just an issue in ESM2. So while we were looking into this, another paper was released, and the authors here actually did a very deep dive into this phenomenon. So a lot of very interesting insights. And for example, one of the insights that the authors had in that paper was that these massive activations might actually be biasing the attention mechanisms. And so, we know in biology that these attention mechanisms are known to capture contacts and such. And so there is a lot of questions that this raises, and I think maybe as a field, we don't poke into these models as much, and maybe this raises some surface area for people to poke into as to what exactly these models are learning if we see these phenomena. And in our case, in the non-language model case, we thought, well, what if we just removed those wacky channels? If we just got rid of them. And so what you see here is if you were to take an histogram of these activations, and you basically drop out the 3 largest values. So now your distribution is much more smooth and much more reasonable looking. But the issue is that if you drop out these 3 channels, the purple is the original prediction of a given sequence, and this green one is what happens after you remove the 3 channels. And so, as you can see, the performance entirely deteriorates once you remove these 3 outlier channels. And so, examine quantitatively. If you look at the TM score to the original structure, once you remove these outliers, the TM score is much lower. Same thing with PLDDT. So this is kind of an interesting thing when you think about it. So there's 1,024 channels in this embedding. You would assume that maybe the information is distributed equally across all, more or less evenly across all the channels if we need every single one of the channels. But it turns out that if you just remove 3 of them, then the prediction ability completely deteriorates. And so this means that it actually might be possible that the information is not spread out that evenly through all the channels, right? And so if that's the case, then maybe we don't need all the channels anyway. Maybe we can just, narrow it down to just a few. And so, yeah, this is, sort of thinking about the implications for compression and thinking about the implications for whether or not all the channels is useful is sort of one of the outcomes of this massive activation investigation. But there's also some other more subtle things too, right? So it might affect the training stability. It might affect whether or not you can quantize and compress the model. And actually, interestingly enough, after we preprinted it, the original ESMFold authors also noted that that was something that they, it was indeed an issue during training for them. And so in our case, we wanted to focus on this compression piece and this piece about whether or not all the data is useful, because as Nathan mentioned earlier on in the talk, this is one of the questions that's very important to us in terms of, we want as much data as possible, but we also don't want to include data if we don't need it to avoid complexity. So what we did was we designed this autoencoder to embed this embedding. So what we have here is on the side, you have the input sequence that goes through the ESM2 and some projection layers, and then that goes to the folding trunk. And the way that Sheep works is we take this latent space that we have just shown is, pathological, has massive activations, and we pass it through this this autoencoder, the Hourglass architecture, and first we do this normalization step that just gets rid of the massive activations, and then we do some projection layers. And the main thing that we want to focus on here is this quantization step, or this middle step. To fix the massive activations, we do this post hoc channel norm and just get rid of that. So we do that first before we pass it through the autoencoder. And in the middle here, this is sort of where we get the actual cheap embeddings from. And so we examine 2 schemes here. So one is this tokenized scheme, and the other is this continuous scheme. So in the tokenized scheme, we use the FSQ method to basically, you can think of it as like snapping the continuous values to like a position in the grid. And then you are basically able to bin them into discrete bins, and each of these discrete bins represents one of your discrete representations. The more popular way of doing discretization in the literature so far has been VQBAE. We found in our experiments that FSQ was a lot more stable. The other way that we look at this is continuous, so without the tokenized part. And so there we just take the output of the encoder, and then we apply a 10H layer to bound the values between -1 and 1. And so this is helpful for things like, being able to diffuse, because usually those values are bounded between -1 and 1. But on top of that, it just also makes it a lot easier to work with when the values are, nicely bounded and you have a reference point. So starting off with talking about some of this tokenized representation. So why do we want to tokenize representations? Well, there's, like I mentioned earlier, some of the motivations we had for Sheep was, generation and for search. And so in generation, there's been a lot of really good works and that achieve very good results by essentially taking an image and learning a tokenized representation over it. And then you can basically apply masking schedules and be able to generate some pretty high quality images. So that's one reason why tokenized representations can be very useful. It also can be very useful in search. So for example, FoldSeek, which is a very popular tool used to search structures against database for structural similarity. That is also done using this discrete vocabulary. And so the search is done in this discrete vocabulary space. So like we mentioned, by working in this space, we can get any representation and any compression we get from working in this latent space of ESMFold, we can get without structured information. We can get just sequence with just sequence, and we can also like explicitly decode out structured information from that embedding. And so this is sort of like how do we make this representation even more useful, right? How do we make it connect even better to existing literature so that we can use it for more downstream things? And so tokenized representations is a very useful thing there. All right, so let's look at the results of this tokenizer. So again, these representations were obtained from just sequence, and we tried the two methods, the FSQ and the VQBAE methods. And you can see that after a certain codebook size, after basically a certain number of discrete values that your discrete representations can take, FSQ actually outperforms VQBAE. And very interestingly, this actually corresponds very well with image experiments as well in literature. And you can get to a pretty good accuracy when you examine it by both the structure, which is shown here, as well as by sequence. Yeah, like it's maybe one thing to highlight here is I think after we printed the papers, people have asked about what's the difference between this and just a normal sequence. So yeah, the normal sequence doesn't actually well, it doesn't just it's only the sequence. It doesn't have the structural information in it, and so it's actually kind of interesting to think about the differential in information contents between sequence and in structure, because once you discretize it, actually you need quite a lot more than just 20 amino acids to catch that information. So there's a lot of very interesting sort of things to think about from this experiment. All right, so the other thing we're going to look at is our continuous side of the experiment. So here we're not actually, we're not going to try to make contact with tokenized literature. We're just going to look at how well we can just compress the information and to what extent can we toss away channels and still keep the performance. So it turns out that actually this embedding space is very compressible. So here we have the channel dimension of the basically the bottleneck of that architecture that we just showed, the encoder architecture. This is the channel dimension of that bottleneck. And on the y-axis, we have the sequence accuracy to before compression. So we put in the sequence, we force it through this bottleneck, and then we decode it back and see how well you can get the match, right? And so the bottleneck basically represents like that's basically compressed representation, right? So it starts saying like how much can we squeeze the passage of information before we can't recover it anymore. So as it turns out, you can actually get down from, originally it started out with 1024. You can get it down all the way up to like, even 8. And you can still get really good sequence reconstruction. That's pretty wild. For structure, similar case, it's a little bit harder. So, the performance starts to deteriorate a little bit earlier, but nonetheless, you can get down to like 16 dimensions from your original 1024 dimensions. And keep in mind, this isn't even including that like contact input as well. This is literally just a sequence representation. And we also tried this with and without downsampling along the length. As I mentioned sort of earlier in the talk, in the beginning when we were trying the diffusion part, we realized that it was hard to fit the entire length of the protein in. And so this is also part of the reason for examining whether or not we can compress lengthwise so that we can decrease the memory requirements for anyone who wants to use this type of embedding for any downstream task that requires like a transformer-based architecture. And so you can see that, yes, even when you apply the length shortening, same thing, it's a little bit harder, but yeah, then you can still compress quite a lot. This is a very similar plot, but it's looking at the RMSD. So it's the all atom, whereas the TM score is the backbone only. And so again, yeah, you can even this is not just it's not just a issue with, or it's not just a phenomenon of backbone atoms being easy to learn. It's for the entire structure. Okay, so then we wanted to look a little bit at function, right? So we use these embeddings for a lot of different things. Getting structure out of them is only part of it. People want to use these embeddings to do a lot of downstream prediction tasks. And so we wanted to see if we were to compress these embeddings, whether or not we can still keep performance on these function tasks. And so what we did here was we took the compressed embeddings, and then we trained a downstream classification head on tasks for the pure benchmark. So all of these are just your various tasks of interest in things that you might want to predict from your protein embedding. And rather interestingly, so the first thing to notice is that the performance drop is much more smooth. It's not like with sequence and structure where it's, there's almost no change until you get to the very end, right? Here it's much more gradual. So, it does actually, it's not a sort of a sudden drop off in performance. And then the other thing to realize is that it's not quite the same pattern between different tasks either. So for solubility, you can still get it down to actually pretty small amounts of channels and get, like not lose that much, performance. But then there are other things like beta lactamase or fluorescence where, if you want to go down to 8 dimensions, yeah, you really are just not going to be able to do very well on this task anymore. And so there are a lot of different explanations for this, and this is definitely the things that we want to look at in follow-up work as well. But one explanation could be that the amounts of information you need representation to predict function is actually quite complex, especially if you want a universal embedding for a bunch of different things. So one way you can think about this is, say you want to compress an image, right? You have an image compressor, and say the structure is kind of the shape of, let's say, the motorcycle in this image. And if you just want to roughly keep the shape of the motorcycle, it's got a wheel, it's got like it looks, it's got a seat. You don't actually, you can get a pretty pixelated picture and still be able to roughly tell that's the shape of the motorcycle, right? But let's say now your task is to predict like how fast this motorcycle is going to go. So that's a functional prediction, right? And so to know that task, it's actually you might have to make sure that you have all the number of pixels needed to look at the name of the motor or something like that, right? So you know exactly how fast that thing is going to go. But then you actually don't need that level of granularity at the other parts of the picture. You only need that for that one part of the picture, and you can pretty much toss out the rest of the picture. So what I'm trying to get at here is that by taking this image compression analogy, it's kind of clear post hoc that of course, like for different functions, you're going to need different levels of resolution for how much you need to, like how much of the data you need to do that prediction, and that it won't be the same for different functions. So this is sort of our takeaway from this experiment. And cool. So now moving gears a little, switching gears a little bit. So The next thing we wanted to look at is sort of going back to the very first thing we noticed of the irregular latent space and of the massive activations. We wanted to see whether or not this autoencoder would be able to quote unquote fix these bumpiness, I guess, in the latent space. So what we have here is the top is the original latent space with its massive activations, and in the bottom we have the compressed latent space. And here we're basically doing a linear interpolation in the latent space. Every single step of the interpolation, we decode it back to the sequence and to the structure, and then we look at whether or not, or we look at the how much it resembles the starting point. So here we have let's do this top one first. So this is the original sequence space, and so we have linear interpolation in the latent, and we look at the degree to which the resulting sequence is the same as the original, and so, or in this case, at the end, the t equals to 1 point. And so you'll see that, you know, like basically you're adding noise linearly in your latent space, but there's pretty much no change in the sequence until and then there's a suddenly this very big change, and then it becomes even again. This is even more stark when you look at the structure, right? So it's almost no change, and then it sort of all of a sudden you're in the other structure zone now. So what this is saying is that it's implying that the latent space is not very smooth. You know, like, and the other thing too is that, you know, we know that most of the protein space is not supposed to be viable. It's supposed to be like, you know, garbage, and that, you know, there are only very little parts of the space is actually viable. But the way that ESMFold is currently characterizing this latent space, that's not the case. So you can basically, you pretty much don't really get to a point where you have a bad structure. Like, you know, it's just like you take a linear interpolation, and in theory, all of this space should be pretty bad, but you still actually get a pretty good structure. And so if you were to look at this autoencoded latent space, you know, you win some, you lose some. So on the one hand, it's a little bit maybe more resembles what you might expect in terms of the actual sort of like manifolds of a protein latent space. But it also means that if we're to look at the naturalness, right? So in the original, because you have pretty much no change, pretty much everything you see is of a pretty good, it maps to a pretty viable structure or sequence. Whereas in the normed and compressed, like the quote unquote fixed version of the latent space, now more of the space is not viable. So this sort of raises a question that I think is, at least in my opinion, I think is still somewhat open-ended. Like, what actually makes for a good latent space representation, right? Is the goal that you want to make sure the entire latent space maps to a good protein? Or do you want it to basically reflect the naturalness of what the protein energy landscape should look like, right? And so, yeah, so this is sort of a bit of an open question as well, but I think that the question that this raises, and that I think you know, is worth looking into more for both us and for the fields, is, yeah, like what is actually going on in the latent space of ESMFold, and how we kind of assume that it's learning biophysics. We use it for a bunch of downstream evals, but you know, the actual latent space and what the fitness landscape that we're using it to approximate is actually quite different from what the real biophysical fitness landscape should be. And finally, this is a similar experiment to the last one, but this one is with the tokenized embeddings. I won't go too deep into this experiment because it's a little bit tricky, because we don't have a mask token. We can't really do a direct masking experiment, and so here we kind of did a crude form of that, where we basically disrupted some of the tokens to see how that maps to changes in the structure space. And rather interestingly, for most of the tokens, it's as you would expect. If you were to disrupt the tokens, you know, no longer are able to get the original structure back. But for whatever reason, there's actually some set of tokens where if you disrupt it, you actually don't lose that much. And so again, this is sort of maybe implying that there's some redundancy in the codebook as well. But yeah, it's a little bit harder to sort of interpret what it means to move and to interpolate in like a discrete latent space. Okay, so a few takeaways from Chief. So one, we saw that the latent space of these protein folding models can, in fact, be characterized as a joint distribution, and that we can actually take frozen decoders and like be able to interpret what's going on here and map it directly back to sequence and to structure. But it's also very compressible. We don't need all the channels. And so this has, different implications. Maybe these protein folding models are over-parameterized. And that, we also saw that you can compress to different degrees for different modalities. And so, maybe different modalities have different degrees of information that it offers as a training as a training data modality. Yeah, and finally, also, I think that this autoencoder hopefully will be just generally helpful to people who, depending on what your use case is, if you're looking for a discrete tokenizer, especially if you want good coverage, and you want to train on data that's for which you don't have structure, but for which a discrete token would be useful. I think this could be a very useful tool for you. And also, if you just need a compact representation generally, there's a lot of practical benefits too that comes out of this autoencoder. Okay, so that kind of wraps up this the findings that we have in this paper. And so as maybe a little bit of like a sneak preview, or we mentioned in the beginning that a lot of this comes from wanting to do these tasks in the middle. So we wanted to characterize this latent space, not just to understand the latent space, even though that is also very interesting, but yeah, we also wanted to use it practically. And yeah, so this is probably going to be work that we will be releasing very soon, but if you in the middle of the talk, I showed a picture of diffusion in the latent space pre-compression, and this is some results that we have from diffusing in the latent space after compression. And as you can see, the PLDTs are a lot better. And after you pass it through the encoder, the space is just also smoother from a when you examine it from the perspective of what happens when you add Gaussian noise into that space. So all of these different things that we did in Cheap actually is helpful from a practical sense. And so this, I think we want to highlight here is that, yeah, there is a bunch of different downstream things that, if you sort of are have a clear idea of what it is that you want to do and what type of tool you need, Cheap can actually, yeah, it turns out that if you compress latent space and make it more organized, you can make the latent space a bit more useful. Okay, so that concludes the talk. Thanks for listening. So yeah, this is a great collaboration with some folks at Berkeley, with many great folks at Precision Design, as well as with Kevin at MSR. And you can check out our paper as well as our open source code. And the weights have also been released, and they'll be sort of integrated into the rest of the code base shortly. So you can just basically pip install and just call the function and be able to get the model and be able to use it for whatever it is that we want to use. Great. So I will leave it at this. And yeah, if there's any questions, please feel free to put in the chats or message the organizers.
Thank you so much, Nathan and Amy. And yeah, feel free to either ask questions in the chat or unmute yourself and ask directly.
Hey, great presentation, Amy. I wanted to ask if you guys ever considered looking at any protein like textual data, and if that was something you ever considered or would consider in the future, and if you think it would offer even more than trying to have like a joint sequence and structure latent space.
Yeah, I mean, that's a great question. And I think that's the answer to that is probably worth another talk. I think it's like there's quite a lot to dig into there. Yeah, I guess, yeah, so two thoughts. One is, yeah, I think we talked a lot about what different types, the information content and different types of modalities. And I think definitely text would have a very distinct, density of information and could probably also decrease the amount of information we needed at the modality. So yeah, so that's very interesting. And additionally, in the work that we will probably release soon to do with diffusion in that latent space, we actually did make use of some textual labels, and so we were able to see that adding that like sort of textual level labels were able to help with steerability. Nathan, I don't know if you wanted to add anything to that.
Yeah, I'd just say stay tuned. Yep.
And I guess another follow-up question I might have, if someone were wanting to use this compressed latent space, maybe to incorporate into some of their like protein design frameworks, do you think this would work in those lower end regimes where you don't have as much data? So then maybe not having, your model over parameterized when you have such low data regimes, like this would work well? Or is this better for building like foundation models and then just compressing that latent space?
Yeah, I think that's a great point. We didn't run the low end experiment, but I think you're right. I think there's a lot of reasons to believe that could work, right? Because then you can basically, if you reduce the number of features, it's a lot easier to build like a smaller neural network from top. So yeah, I think there's a lot of reasons to believe that to be the case. It probably would depend on the downstream task, as we saw in the function-related benchmarks, right? It wasn't always so uniform in terms of, or it wasn't the same for every single task in terms of how far down you can compress before you sort of are no longer able to perform as well on the task. But I think that's a great point. I might also add that I think from just loosely from probing around, I think that tasks that relate to structure were easier to compress, given everything that we just discussed about how structure is easier to compress than other functions. The things that I found to be a bit harder is especially if you looked at beta lactamase tasks, like that the model wasn't doing so well. All of the times, it's like one very small variation, like mutation level difference, can create a big difference in the outcome. That's the kind of thing where I think, compression is a little bit harder to handle that case, right? Especially if you're also compressing along the length dimension, then you lose some resolution there.
Awesome. Yeah, that makes a lot of sense. Thank you so much.
Hugging Face waits repo show 404. Yes, I will. This is literally the top of my to-do list after the ICO deadline. So I will fix that. And the plan was to make it very easy for everyone to use. It's just number one had to go through some approvals, and number two had to go through some pain with the ICO deadline. So can this be used to compare two different language models? That's actually also a really great point. And again, I think the answer to this question also kind of opens a whole can of worms in the mechanistic interpretability space, and could probably be a whole paper. But there's a lot of works that look at to what extent the weights of language models contain information, and looking at language models as sort of a compression of the data set, of the internet scale data set. And so the point I'm trying to make here is I think there's actually quite a lot of literature that poke at different angles to this, and the full answer will probably be quite complex. But I think in short, I think you're right that you can probably use this as a tool to answer these sort of more deeper, more mysterious questions about how language models work. Are there any questions that maybe people are not able to ask during this talk? Please feel free to reach out to me or to Nathan and to other authors. Our emails are all fairly easy to find on the internet. Rather than compressing the embeddings, why not trying at ESM2 using embeddings with significantly lower dimensionality? This is also a great point. So one thing that I wanted to note is a big part of the motivation here is to avoid retraining ESM2. So everything that we mentioned, like the whole massive activations, it's actually very easy to just retrain and just literally add a channel norm, and then you can fix the massive activations. But a lot of the motivation here was we mentioned earlier in the talk that we wanted to make use of the trend in other AI domains where you can do a lot with pre-trained models, like in robotics, you can do a lot with the information that's been captured in ChatGPT. So we wanted to not look at fixing ESM per se. We wanted to look at this more broader paradigm of like, how do we extract information from these models and how do we make them useful? So that's why we didn't go and, sort of retrain ESM2. But if you just wanted to get a compressed embedding, and if you just wanted to, and you have the compute, to like retrain ESM2 and had the time and had the interest, then yeah, that would be another way to fix it. But we wanted to look at more lower cost ways of doing this.
Yeah, and I think that since now that Amy's diagnosed the problem, it would be definitely a lot more straightforward to just train from scratch and potentially alleviate that. We have run some small scale experiments, just trying to train PLMs from scratch with like smaller embedding kind of bottleneck dimensions. And I think like for the same reasons that over parameterization is just good in general, training like a big expressive model, that has like many paths to degeneracy and then compressing it is potentially easier to do just from what we've seen than trying to engineer that in directly.
Yeah, I think that broadly, like in AI, there's this paradigm towards, just sort of viewing the pieces of big pre-trained model weights as tools in themselves, right? Like you can take different layers from different parts and then use it for different things. And so, to Nathan's point, I think there is also potentially some use in, you might be able to get some performance gains or have more flexibility by training a big model and then, compressing it post hoc. Should we try to recognize spunky activations in facial recognition neurons of LLMs? Yeah, probably. I mean, I think that there's been other parts of LLM literature, not very big parts of the literature, but there are some discussion about how these massive activations can prevent you from quantizing the model, right? Because now your numerical range is very much expanded, whereas otherwise you can use like a much slow, like now you have to use, let's say, like floating point 64 because you need all the numerical precision. So I think like in short, probably you probably should regularize. And part of this also I think comes from the fact that we're so used to using layer norm instead of other, like we just kind of use layer norm because that was what was used in the original transformer paper, but layer norm it does not fix this sort of like the fact that some channels have really big values. And so maybe one way to do this is to sort of alternate a little bit between layer norm and between channel norm. That could be a way to maybe look at this, but this is not in terms of how it affects pre-training dynamics, it's not an experiment that we run. And so I don't want to like comment on exactly how that's going to turn out.
Okay, if we don't have any further questions. Thank you so much, Amy and Nathan, for an amazing talk, and thank you everyone for joining. And look forward to seeing you guys at a future seminar.
Thanks for hosting it.
Thanks for having us.